Welcome to the Wired for Success podcast, your show for ambitious, mission-driven entrepreneurs who want to build million-dollar businesses without sacrificing their health, relationships, or happiness, who understand that in order to build their empire, they need a solid foundation, and that the more they are thriving, the more their business is thriving too. This show brings you the very best from science, self-development, and entrepreneurship to set you up for sustainable success. Hi, I'm Claudia Garbutt, the scientist turned mindset and high-performance coach for entrepreneurs, and I help you get wired for success.
Hey everyone, and welcome back to a brand new episode of the Wired for Success podcast, where we talk about all things science, self-development, and entrepreneurship that help you get to that next level of success in your life and business. Today's guest is one of those people who sits right at the intersection of science, technology, and real-world impact. Dr. Nisim Perets is an Israeli neuroscientist and biotech entrepreneur working on something that sounds like science fiction, but is rapidly becoming science fact. Patient-derived brain tissue grown in the lab on a chip designed to predict how a specific human brain might respond to a drug before it's used clinically. He has published multiple high-impact papers, holds more than 10 medical patents, and was part of the scientific founding team at Neuroscience Biologic, developing nanoparticle-based therapies aimed to recovery after severe spinal cord injury. In 2021, he founded Itay&Beyond, a company with the mission to use brain organoids to accelerate precision medicine for neurological and psychiatric conditions like autism and epilepsy. Now, this sounds super fascinating to me, and I can't wait to learn more. Welcome, Nisim, and thank you so much for taking the time to be on my show today.
Thank you for inviting me.
I'm so glad we made this work. Now, Nisim, the company name is Itay&Beyond. Can you share the story behind the name and what makes your mission so personal and special?
Yeah, so as you said, at 2021, I established Itay&Beyond together with one of my partners, Shmulik. The story started when I finished my PhD in neuroscience. I was studying new methods for drug delivery, which means to carry drugs to the brain safely using nanoparticles. And I had a few successful papers in high impact factor, and also the Tel Aviv University did some PR on my work. And it got to the years of a successful high-tech entrepreneur named Shmulik. And he came to me to Tel Aviv University, and he told me his personal story that he had an autistic son, low-functioning autistic son named Itay, and he and his wife traveled around the world to search for not only for a treatment, but for a diagnostic tool or for a way to match existing treatment that will benefit his behavior, not only the autistic behavior, the severe core symptoms of autism, but the ADHD, the what we call like the secondary symptoms, and basically he found nothing. And they did a complete genetic test to Itay, and it came out clean, which means that the boy is severely autistic, and the genetic test is clean, so no one can do anything, and also the MRI and everything. So after I finished my PhD, he came to me and he said, let's work together with his funding and my ideas to bring something new, a new technology, a new way to think of not only autistic spectrum disorder, but also other disorders as well, neurological and psychiatric disorders. So we established Itay&Beyond. The purpose was to put the name of Itay in the front and to say, okay, we're starting with a single patient and go beyond to different patients in autism and different pathologies beyond autism. And this is exactly what we did. So that's the beginning and the story behind this very unique name.
Oh, I love it so much. It's such an inspiring story. But I wonder, how do you hold that kind of emotional mission without letting it turn into unhealthy pressure? Because it's very personal, right?
Yeah, so luckily, first of all, Shmulik and Osna, the mother of Itay, they are very intelligent people, okay? So they understand that treating autism and finding a treatment or diagnostic tool, a new diagnostic tool for autism is a very difficult mission. So we put it at the beginning, as a condition, to understand that if we want something successful, we need to think, not only on the personal story of Itay, because that would also would make the family invest a lot of money on a single case, which is not very clear how we can decode it. What I explained at the beginning as a scientist, that I cannot work only on Itay anyway. To know what is wrong with Itay in the neurological way, I need control patients, and I need patients with autism that is, let's say, genetically characterized. And then I can compare Itay to the control patient and to the patient that has like a Phelan-McDermid syndrome, which is a type of autism, but is very genetically characterized in a specific mutation in a gene called chunk3. And then I can map it and to understand what is, what is the specific pathology of Itay and other patient with what we call idiopathic autism or multifactorial syndromes of autism. I need to start mapping it to know what is considered to be control, to be healthy, and then to know what is the manifestation of the problem in a well-characterized a patient like Phelan-McDermid syndromes or grade 2B syndrome, and then to see where Itay fits in all this map. And this is exactly what we did, and that's basically the vision that carried us. So it also took the story to a scientific level and not to a personal level. And not only scientific, we talk about a company. A company needs to be successful. You don't want it to be, a endless hole of money that you just throw in and throw in. You want something sustainable that will carry on, regardless of of the funding that the private family have or of a few families. You want something with incomes, you want something that will grow, because it's a long journey. It can take a year, it can take 5 years, and it can take 10 years. So it's something that you need to think of when you're establishing the company to make it sustainable, to have a value that other people, if it's VCs, if it's customers, if it's anyone, will see value in it to help you with your journey. So that's the way from the beginning to work on it.
Yeah, that makes total sense. You have to make it translational so that other people can use it too, and you're part of the puzzle, like a puzzle piece if you want. Now, talk to me like I'm a 5-year-old and explain what your company does. So for someone listening while driving a car or making coffee, what exactly is patient-derived organoid, a brain organoid on a chip, and what should people know about it?
So first of all, it's a cutting-edge science, but it's a cutting-edge technology, but it's not something that we or I invented end-to-end. Okay, it's a combination of a few things that were already known to science, and we just pull them together to a story or a technology from an end-to-end, and I will explain. Basically, our technology is based on the ability to take a single cell from a patient, even non-invasively. In Itay&Beyond, by the way, we do it from urine sample, just take urine sample from patients. And then in urine sample, for example, you have cells that you secrete all the time from the kidneys, from the liver sometimes, and the body is cleaning them and they're still alive and you can use them. So once you have a cell, if it's from the skin or from the urine or from the mouth, whatever, we have today the ability to take this cell and to basically reprogram it to become a stem cell. A stem cell is the type of cell that we're starting our life with. It's a fertilized egg, or a cell that can still multiply and become other types of cell in the body. Our brain, our skin, our liver, our lung. Okay, so once you take any type of cell, like let's say a skin cell, and you reprogram it back to become a stem cell, then you have stem cells of the patient. Okay, and you can do with them a lot of things, not everything that you want, but a lot of things, including to make them now become gradually a miniature living brain tissue, okay, which is about the size of this, okay, this size, and we can see them. We can work with them in the lab. You don't need a microscope to see them. You can touch them also, and then they contain, let's say, like 10 million cells, neuronal cells or brain cells of the patient, and you can work with them. You can test drugs on them. You can test their activity, electrochemical activity, because the brain has an electrochemical activity, so you can measure it. And what we decided to do, and I have it in the picture in here, is to take the lab-grown brain tissues, the brain organoids, and basically to put them on a multi-electrode array, a type of array that can record and stimulate the brain activity, the electrochemical activity of this tissue, and it's connected to a computer, so we can measure it all the time. That's the technology end-to-end. So basically, we take a urine sample from patients or from healthy volunteers, and from this, we gradually grow hundreds of brain tissues, put them on a multi-electrode array that is connected to a computer. And once we have this, like we are doing a lot of other crazy things, which I can explain.
Okay, so once you have your mini brain on a chip, then you can start your drug test. Is that correct? Or is there...
Exactly.
Okay.
Exactly. And the question was, like, what kind of drug testing do we want to do? What would be the readout that we want to achieve? And I'll give you an example. In the academic world, and also in early, we call it preclinical pharma studies, we are using usually mice as an animal model to test drugs and to develop drugs. Okay, mice, rats. It's rare to use another type of animal. Sometimes we use fish, and in an extremely rare case, we're using pigs or even monkeys. But the regulation, the FDA, the EMA, they're all getting away from it right now. Yeah, it's a trend now to stop using them. And the reason is that animal models to test drugs, especially for neurological and psychiatric disorders, are considered a poor predictor for the efficacy. Okay, so a lot of drugs were proven successful in the preclinical studies on mice and rats, and failed completely in the clinical trials in the patients themselves. And then it generates a lot of problems. The first problem, that you're using animals just for nothing, and it's too bad to do it. Second problem is that you're spending lots of money and years of research of something that is based on a science that cannot predict your success. So you basically have a low chance of success. The third problem is that you're misleading drugs that didn't work on mice but could work perfectly on humans. Okay, so you have all these problems, and with this you go to the clinic and trying to solve problems like autism, like Alzheimer, like dementia, Parkinson, all of this. So it's like in a bottleneck that it's not only helping you, it makes more harm and damage than help. Why are we using mice and rats? There's a reason for it. And the reason is that you can produce functional readout. For example, if you take a drug and you give it to a mouse that was genetically modified to have Alzheimer, for example, we can do it, or autism also, a specific type of autism. And then you can give it drugs. So you know how a normal mice it needs to behave in specific environments, like social interaction or memory or swimming or whatever. And then you give the drug, and if it improves the phenotype of, let's say, dementia or Parkinson or even social interaction in autism, you have your functional readout. That with this you can go to the FDA or the EMA or to start clinical trials in human. Without this, it's very hard to know if you have any lead whatsoever for your drug. And that brings me back to the problem that we had, or I would say the question that we had in Itay&Beyond at the beginning, because we had this miniature, beautiful brain organoids in the lab, okay, and we can test drugs on it. But how do you know that a drug benefits the autistic syndromes or any type of change in just organ in the lab. And that's when we took the extra mile. When there was a paper published around the weekend, one of the weekend at the beginning of the company, we just opened it, that I read and I was very fascinated from a new type of technology. They took rats, they took out their brains, okay, and they dissolved it into single cells. They put the single cells on multi-electrode array, and they used some kind of a mathematics that they knew that called closed loop reinforcement learning to teach the cells in the lab, okay, on top of the multi-electrode array, to activate a computer game in the computer, okay? So they just learned it. So you had like a few rats' brains, neurons playing ping pong against the computer in a lab. So for someone out of the scope, this could be like, wow, I would say science fiction. But for me, it wasn't science fiction because I knew everything, and they said, this is cool. So and I read the paper, I read the mathematics of it and the methodologies and everything, and I said this can be possible. There are a few challenges, but it can be possible to translate it to the brain organoids that we have in the lab. We already have brain organoids. What we need to do is to put them on a multi-electrode array, teach them to play computer games, and then if we will have luck, and in this part you need a lot of luck, we will see different in the performances of the brain organoids, of the lab-grown brain tissues derived from urine of patients with autism, rather than the patients of control group, for example. And we tested it, and it worked. Not only that we could basically generate a closed loop reinforcement learning from the brain organoids to the computer, which took a while. It's not just that we saw a paper, I read a paper, and it works in the lab. It's not like this. And there's a difference between rats. Yeah, there's a difference between rats, few cells in a dish, to a three-dimensional brain organoids derived from human with reprogramming. There's lots of changes that needed to be done, but we succeeded. And then we saw differences in the behavior, the electrophysiological behavior that was translated into a performances in the game between the patients of the brain organoids taken from patients and from control. And that was the moment that we realized we can use brain organoids and extract functional readout from them. And that's a game changer for a pharma company and for drug discovery platform company. Then you don't need the behavior of the mouse anymore. What you have is a behavior of a brain tissue derived from the patient itself that is going to get eventually the drug that we are developing in the lab, or that we want to repurpose from like epilepsy to a specific type of autism, or this kind of repurposing.
It's so cool. Can those brain organoids solve other problems as well? Can you teach them to solve other problems? I know that sounds crazy, but
no, it's not crazy. We actually have a lot of, I would say, a lot of simulations. What we're trying to simulate is a behavior based on memory. So we have a memory test based on attention. So we have attention test where the signal is coming from, they need to respond. And there, as I said, it's not something that we invented. There is a field, a very new field at the cutting edge of science that called organoid intelligence, and people, the smartest people in the world, trying to understand that organoid intelligence may solve a lot of problems in the fields of AI, machine learning, especially in the dynamics that requires high efficiency and efficient energy consumption. Okay, for example, we just joined a program named NVIDIA Inception. It's an Inception program that NVIDIA has established for startups that develop cutting-edge technologies. And together with them, we can work on new technologies that will improve neuronal networks, which is the mathematics behind all the machine learning and AI, and efficiency consumption, especially when we are using brain tissues. Brain tissues have enormous ability of efficiency in energy consumption. Our brain can do really, million of calculations in a second in an energy consumption that is less than a lamp on the wall. So that's the idea. You can think of it when you drink your coffee in the morning. You have enough, and it gives you enough energy to do all the calculations for a few days even. So imagine computer would be that effective. So that's one thing that brain organoids can in the future, and I believe the very near future, can do.
Now that's so cool. And I think researchers have also trained organoids to modify their responses to stimuli. Is that correct? So
that's a part of the game.
Yeah, that's so cool. What are the limitations of this method, and how does it compare to different corners of the synthetic biology fields, like nanobots, for example? They're kind of like in a different corner of this entire field. They are programmable as well, to a different extent, though, right?
Yeah, you're referring to xenobots, the cells that can move and then okay. So I will explain. First of all, it's a different field of biology and of science. I will start with your first question, the limitations of these technologies. So first of all, it's a new technology and it always developed. I just want to say that it was very hard for us at the beginning to generate brain organoids with electrochemical activity. It wasn't such an easy task. Our first brain organoids that we developed were beautiful. They looked amazing. And then we put them on the multi-electrode array and there was silent. There was no electrochemical activity like a brain. They behaved like any other tissue, but not as a brain, of an encoding information machine. And it took us about a year and a half and a lot of persistent courage and belief in the way both of our team and me and Shmulik and the other investors, for us to tell them confidentially it can be done. We will generate electronically functioning brain organoids. Not only this, they will not only be electronically active, they will encode information and they will be differentiative between patients and healthy control. That's like huge mission, and we didn't know if it's biologically feasible. We didn't know, because I just want to remind you, we take urine sample, we reprogram cells, and then we generate synthetic brain tissue. Nobody promised us that this synthetic brain tissue would have nothing to do with the patient at the beginning of the way, okay? And it worked. And I always say, when God loves your way, it works. Like when it doesn't, no matter what you will do, it will just not work. So there's a lot of limitations because, first of all, we're talking about a synthetic brain, like lab-grown brain tissue. It's not a real brain. It's not like it doesn't have all the capacity of the brain, all the areas, all the connectivity. But still, we are programming it to become frontal cortex, the most important area of that is related to a lot of neuropsychiatric disorders. And we also have the ability to generate few other brain regions and then connect them together to play with this. But it's still the beginning, so we have lots of challenges. Another challenge, and people have asked us about it, is how similar, if I grow 10 brain organoids from a specific patient, how similar they are. And there is a variability. And I always say it's that, of course, there will be a variability, but when we take a single patient, and we already have tens of patients and thousands of brain organoids that we grew, and we see it, then they're different from one another, but they are very close when they're taken from the same patient. And they are closer from different patient if they are control group to another patient which has autism spectrum disorders, for example. And this is what's important. The important thing is not that they would be exactly similar, but that the variability between them would be manageable. We can say, okay, this is in a chance of like, let's say 80%, not only that it's healthy, but in a chance of 80%, it probably belonged to, to James, for example, because we see some features, and these brain organoids look and behave more or less the same as this James person. And they're not like of Jamie, which is autistic, that's for sure. So that's what we can do. And regarding xenobots, I know the field quite well. I would say it's a fascinating field, and it has different questions. I don't know if they're taking it to first of all to I would say drug development or drug discovery. I will just explain for some people that probably didn't hear. Xenobots is the ability to take cells. I think they're taking it from specific animal. I don't know how to say it in English.
Xenopus
Yeah, exactly. I'm sorry, English is not my first language, so there are words that I don't understand. And they're taking it, and then they manipulate it to become like some kind of a biological robot. So it can move in the area, it can learn to do things. It asks different questions about, I would say, the basic intelligence of biology. If a single cell can be intelligent, if they work in a group, how they communicate. So it's a different field. It's fascinating, and maybe one day I will learn more about it. But it's not the same as brain organoids that you grow a lab-grown brain tissue from patient to test drugs and compounds.
Yeah, and I think you alluded to this before, but in general, so do you think we can and should use human neural tissue as computational hardware?
Yeah, I think that in the future we will have no choice. That's the truth. And I will explain why. And this is my point of view. It has nothing to do maybe to other people's point of view or other experts in the field, but this is my point of view. So and my calculations, by the way. When we are using AI, specifically machine learning, and the things that became very common, the energy consumption of it's huge. You cannot even comprehend how much energy that ChatGPT and Gemini and all of this and the other programs are consuming in each prompt, in each request, and it grows exponentially. A lot of people are using it right now, and it's not like asking Google what is the capital of London of England. It's not the same. It's a different computational method that based on mathematics that called neuronal network, and it consumes a lot of energy to answer your question or to generate a photo. And the more that we will use it, we will have, as a humanity, a very big challenges, because the chain of I would say the natural chain of generating the clouds and the chips and the hardware for it is growing more than nature can provide us for, the minerals that we need to start generating the program, and the metals that we take from the earth. I'm not talking about like some kind of an acclim change or environmental crisis. It's not me. I'm very pragmatic. What I say is that there will become a point that if we will want to benefit the AI and machine learning as we are today, it would be rather a restriction of use, because the energy will not match, or a new method to have better energetic abilities. So we can extract better energies from nature, or on the other way, we can make the software more energetically efficient. And if we will use, I would believe, brain organoids or stuff like this, based on a biological inspiration, nature do it perfect. I mean, it's hard even to imagine how efficient nature is. Then we can continue developing. If not, then we're going to have a problem. And so I think that At the end of the road, we will not have a lot of choices, but to start thinking of nature as a source of inspiration for the things that we are doing.
Yeah, nature has solved the energy problem pretty well, I'd say.
Yeah,
Right?
we are a part of nature. We are not separated from it. Like we are natural creatures, and maybe nature wants us with our intelligence to go on this road. I don't know. It's very philosophical, but I think that now that we have the ability to grow brains in the lab and to really understand the way that they're performing computations in this low energy consumption, we are on the right way. To better use nature and not abuse it. That's my point.
Mm. Do you personally have any ethical concerns when it comes to lab-grown brain tissue? I mean, it might sound like a weird question, but many people believe that consciousness is a direct result of brain activity. So do you think that lab-grown brain tissue could be or become conscious at some point?
My first answer is no. It's not. It's not. No. It's not that they can't. It's that I don't think that there's a problem, and I will explain. Okay. Yeah. It's much more simpler than this. I think that if we would so concerned about using conscious creatures, then we would never use mice and rats. Okay. To begin with. So that's I think that's just a trend and to think of things that might happen when I used rats and mice as especially in neuroscience biology, for example, that you make a healthy rat a complete spinal cord injury and then you need to fix it. Okay, and if you will not do this, then people that had injuries or severe spinal cord will never have a treatment. So you do it, and you don't ask yourself, is it ethical or not ethical? You know that it's not ethical, you know it. And you know that it's, in the limitation of ethics that you allow yourself to do in order to have other benefits. This is something that if you're not aware of it, then it's a problem. It's a logical problem that I didn't face because I knew that they have conscience in some level, that they are suffering. They are natural creatures. I believe that the brain organoids that we will have, the few million cells, will not have the same abilities as a mouse or as a rat in the perception of pain, in the perception of fear. It's not there. And even if they will have, I rather them to have it rather than a rat or a monkey. Okay, so I don't think it's an ethical issue. I think it's the other way around. If we can control it and if we can grow lab-grown brain tissues with enough abilities to help us understand about our biology without harming or growing rats, mice, monkeys, rabbits, and damage them, we can leave them alone and play only with the biology of it. So of us, I think it's much more ethical.
It's such an interesting question where consciousness comes from and how it evolves and where, is it just little steps that accumulate? Like, are there creatures with very low consciousness and it increases as complexity increases? Or is there a certain cutoff where you say, okay, below this threshold, they're pretty much not conscious, and then after this threshold, they are? And what's that step? And why don't we know?
So it's a philosophical question that goes back to ancient Greek when they ask, how do I prove that you have conscious? I know only of my conscious. You can look very complicated to me, but I don't know if you have a conscious. You may be a bot speaking to me right now. And in our reality of deepfake and AI, we're becoming very close of really blurring the idea between complexity and conscious. I don't believe that if I will have a prompt right now of Claudia that looks like Claudia and speaks to me like the ChatGPT, it's conscious. Not yet. I don't know. Maybe it isn't. I don't know. But I can have exactly the same conversation with a digital Claudia right now. Like, I mean, in 2 years from now, it will be very possible and very easy. And still, it will be very complex, much more complex than an ant or a bee. But still, I'm not sure it's going to have consciousness. I'm not. I'm focusing on curing neurological disorders, and and developing cutting-edge technologies and bringing new science to the world. These questions, I don't know. I mean, they are very interesting, but I just go around them. Yeah.
It's hard. How do you see the cost-benefit ratio evolving between your method, which sounds kind of expensive, but I don't know, and other standard lab models that we already discussed, like mice, for example, or just standard tissue culture?
So first of all, that's a very interesting question. Let's talk about the price and the value. Okay, right now, you're right. Having a lab-grown brain tissues is quite expensive, but I want to tell you that it's not much expensive rather than to have an animal facilities. Carrying the It's not that expensive. I think it's more or less we came to this ratio that to grow lab-grown brain tissue, it's from patient doesn't cost that much because you're not looking about a specific mouse, right? You need the facilities, you need the household, you need the food, you need the environment. They have restrictions. Not every animal facility or any zoo can become a preclinical site for testing drugs. You need, for example, the air conditioning, the air replacement to be 7 times, I think, in a minute. And you need their food to be very specific. So it's very expensive. It's not that cheap. And then we have the other way that having lab-grown brain tissues 5 years ago was, I think, 4 times more expensive than now. Okay, now, we are doing it quite easily and much more efficient. And it's still not that cheap, but it's much less expensive than it was a few years ago. And it will be much cheaper as we go around the road. We believe that Itay&Beyond, our company, will have scale up just the next year. So we will have even cheaper process. That will be very cost effective. And then you have the other question of what is the value. So if mice and rats would give high value, there was no need to develop other things. We would just use them and we'd have a cure for Alzheimer and that's it, or for autism and that's it. Their value is very low. It was proven throughout the years. So we don't have any problem to say that our system can be very beneficial, especially when we're talking of personalized medicine or drug testing before clinical trials for what we call study design. Imagine that you can know which of the patients is responsive to a drug before you sign him into the clinical trial. Okay, just to make a very difficult stories. A lot of the companies and the clinical trials can fail from 2 or 3 patients that didn't respond. Okay, and then the statistic fails, and then the drug fails, and then billions of dollars. It's like this. It goes down to a specific patient, and if you can choose them from the beginning to put them on the trial and to make a better study design, it's becoming very valuable for pharma companies and for hospitals, for example.
Speaking of drug delivery, so once you've identified a drug in your system, you still have the problem, the challenge that it would have to pass the blood-brain barrier in the patient, right? So how do you go about this?
So easily, the first step is we do repurposing, which means that we choose approved drugs, FDA usually, or other regulatory agents like them, and we screen them for autism, like for example, or for specific types of epilepsy. We don't have just autism. We have epilepsy here and some patients of other disorders such as schizophrenia. So we screen them. We know from the clinic that they're working, or that they are working for neurological disorders because they have the chemistry that they can cross the blood-brain barrier. And what we test here is just the responsive of the specific patient. Once it gets and meets the specific biology, what will happen if it will respond or not respond. And For example, one drug that we found that crossed the blood-brain barrier, and we tested on several patients in our lab that called suramine. The drug is called suramine, and was originally designed for a parasite, a stomach parasite, and it's given in Africa. We found that it can be very helpful for several patients in autism, including Itay. And how did we get to this rare drug? Because it was already tested in clinical trials for autism before, I think a decade ago. The studies failed, but the specific subpopulation responded. So the statistic was not good, but we saw that, and people talked about it, that from the low, I would say low autism spectrum disorder patients, some of them even started to speak or to increase their social interaction, which is amazing. But the study was failed from different reasons. And then it went on again, and again the results were better, but not so convincing. And when we tested the same drug in our lab, we saw that we can detect from the beginning who will respond and who will not. That story is not so optimistic so far at this point, because the company it's very hard for us to get this drug, because it was originally manufactured by Bayer and they stopped manufacturing it because it's not in the pipeline anymore. And it's very hard to get this drug today for clinical trial, to say, okay, now we will take the children that responded and we will do the clinical trial again, only with them, and we will show that it works. And as we speak today, we are already, I think, after 8 or 10 months of just trying to get a hold on this specific drug in an approved way to start clinical trials. We have the doctors, we have the hospitals that want to do it, we have the regulation. We don't have the drug, sometimes it's a story like this.
Yeah, that's a pity. But so you are working with already approved drugs instead of screening for new compounds. Is that
We do
right?
both. We do both. The new compound, we have the delivery issue, and we can test. And usually the new drugs, the delivery in animals quite well. So when you give, for example, intravenous, which means to the blood, the compounds, you can detect if it has crossed the blood-brain barrier of the mouse or the rat. For this, mice and rat are pretty good. Their blood-brain barrier are somewhat similar to those of humans, and for years people can extrapolate just the crossing of the blood-brain barrier from mice and rats. So if we have a hit of like a new compound, and we want to know if it will cross the blood-brain barrier, we don't do it in our system. We just pay a CRO, which have several mice and rats, and then we can test if it's crossed the blood-brain barrier there. And we have another method. If we want to take a very effective drug and to make it cross the blood-brain barrier, then we have the knowledge to do it. We can, have a delivery system, and it's a completely different science. But luckily, I have again from neuroscience biology that you mentioned at the beginning for spinal cord injury. I was basically the expert of drug delivery there. And especially drug delivery for the brain. So I specifically my background enables me to know which drug is suitable to cross the blood-brain barrier, and if it's not, which delivery system may help it. So it's like it's a challenge, but it's not the biggest challenge. Yeah, what we say, it's not a atom science or nuclear engineering. Yeah.
What's the biggest challenge then, if that's not the one?
No, it's a challenge. I'm just kidding. But the biggest challenge is to have a drug that eventually will work on patients. Okay, like to cure or to help autism or epilepsy or Alzheimer. It's quite a challenge. And though we believe at Itay&Beyond that we are on the right way to help patients, maybe not to cure, but at the beginning to bring a new generation or to screen drugs that people haven't thought about them and they can help specific patients, we are on the right way, but there's a long way. It's very challenging. I'm not.
You have to stay optimistic though. And
you have to be optimistic for this field and to be a CEO and
Absolutely. And as I know in science, most of the things that you try don't work, or they don't work for years before you get them to work. So yeah, you have to keep that positive attitude. And over the years, I would say that in cancer, for example, we have seen that precision medicine has made huge progress. In the brain, though, it kind of lags behind. So from your perspective, what makes the brain uniquely resistant to precision medicine?
So that first of all, that's a very good observation, and I want to take it even further. The precision medicine is very successful, as I know it, specifically in oncology, in cancer, and over there the field is very can bring much value, of which a medicine can work with the specific type of cancer. There are also other precision medicine that are much easier, for example, skin, trophic medicine, etc. And the common between them, what is shared between this field is that they have a very good testing model. So what you do in precision medicine is that you take the disease or what is mimicking the disease, and you can, of the specific person, you can grow it in the lab and then screen on the tissue or on the organoid, for example, different compounds, and then responsive, not responsive, and you give it to the patient. In cancer, it's worked very well just because you can take a biopsy of the patient from the cancer and do it, and then you have a fantastic way of precision medicine. Obviously, neuroscience, you cannot take a piece of the brain and test it, but what you can do right now with technologies like ours is to have a miniature living brain tissue of the patient, or a few of them. That's something that was never done before. So I expect that this field of precision medicine for neurology and psychiatry is going to skyrocket at the few years. Not only this, we at Itay&Beyond have a few collaborations with hospitals, including Schneider's Children's Hospital, for exactly for this purpose, not on autism. On autism, we work with Hadassah, a different hospital, but with Schneider, we work on epilepsy. And they give us specific patient. At the first step, it's a validation. So they give us several urine samples of patients that are responding to a specific drug, and they have severe epilepsy. In our lab, we see at the first stage if our system can detect that this specific patient respond to this specific drug. Okay, this is the first step of the project, which we are doing right now, and we're getting promising results. The second step is to take the non-responsive patients, and after we prove that our system is validated for epilepsy, with the ability to test which drug would suit the patient, and it correlates to the clinic, we take the non-responsive patient and start testing drugs on them for precision medicine for epilepsy. So that's something that is going on right now.
Can't wait to see where this is going. Now, you also mentioned that you worked with nanoparticle therapies for spinal cord injury recovery before. Can you share the basics of how that technology works and what results you were able to achieve?
Yeah, so that's a different story. It started in my PhD. Also, the story begins when I started my PhD in neuroscience. After I did master's degree in computational neuroscience, I went to translational neuroscience, which basically explore the abilities of different type of stem cells to regenerate tissues in the body, not only tissues, but the brain and the central nervous system. And I worked with specific type of cells that called mesenchymal stem cells. They are derived from the bone marrow, and they have unique properties that can help tissues being regenerated. Okay, and what I found, and again, it's hard to say I found, because then I would get a Nobel Prize on this, but I was among the first people who talked about it, and I had lots of discoveries in this field, that when you take the exosomes of the stem cells, which mean exosomes are a unique nanoparticles. They imagine like a small balls, okay, a vesicle we call it, and that contain a specific information that is secreted by the cell. For example, if it's a mesenchymal stem cells, it secrete a lot of exosomes with a specific molecules such as RNA molecules, proteins, combination of a molecule in each ball like this, in each vesicle. You have like around 800 different molecules, but they're derived from the mesenchymal stem cells, and they are used in nature for cell-to-cell communication. It's like an envelope that the mesenchymal stem cell sends to the tissue and helps it regenerate. And this happens in the body all the time, from the immune system to the injury site, and from different organs as a way of communication that everything is okay. There's lots of exosomes-based interaction. So in our body, we always have these little bouncing balls from cell to cell. When you take them from cell, when you grow cells in a dish and then you make them extract a lot of exosomes, and you take just the exosomes, first of all, they have amazing ability to cross the blood-brain barrier easily. Though they are big, they have this machinery that helps them. And then you can use them for two things. If they come from a cell with natural regenerative properties like mesenchymal stem cells or immune cells, then their exosomes apparently carrying any important information to encourage the tissue to regenerate. So you don't need the mesenchymal stem cell. You need it for something else, but you can convey most of its effect just by using the nanoparticles. Okay, so you can use it as a I would say cell-free cell treatment. Okay, or a cell medicine, but you can also do something more clever and to take a drug that you know that can help a specific disease, to load the exosome, this ball, with this drug, and to use it as a drug delivery system for a specific area in the brain, in the body. And then you will get a synergistic effect. You will get the natural properties of therapeutic abilities of the exosome together with the drug that you use. Okay. One of the projects that I did in the PhD was a regeneration of the spinal cord injury. It was a collaboration between our lab of Professor Danny Offen that I did at PhD, and another lab in the Technion of Professor Shulamit Levenberg, another professor that works on spinal cord regeneration and tissue engineering. Okay, and in this project we tried to use exosomes loaded with a specific molecules that knows to help neurons regenerate after injury. And what we saw that rats with a complete spinal cord injuries that can no longer walk, and then zero, I would say, a spontaneous recovery, started to walk and move their legs and even gain functional walking about 4 to 8 weeks after the treatment. And not only this, the treatment was not invasive, it was given to them intranasally from the nose. And we published a huge paper that had lots of success of showing that exosomes from mesenchymal stem cells loading with a specific compounds that called siRNA for PTEN, never mind, that together they can go from basically travel from the nose to the spinal cord injury and promote regeneration that will end up in functional abilities again. And we did all the science behind it, and it was incredible. And then basically Neuroscience was established on the base of this research and of these results. And Neuroscience today is an Israeli company listed in the Canada stock market and also in Germany, and they are heading towards clinical trials in humans to get the FDA approval. Also in Israel, they are doing they're studying compassion treatment, it took them a while to establish, the regulation and all around this. But the science is exactly the same science that I did in my PhD, the 2019.
Oh, that's so amazing. Now, I know a lot of scientists who really love their research, but who hate any form of sales, marketing, etc. So they would never start their own company. Did you enjoy entrepreneurship straight away, or was it a bit of an acquired taste?
That's a difficult question, and that's a good question. I don't know, actually. I don't know. I'm still thinking about it, but I still don't know the answer. But there was something different about specifically my academic career and the research that I did. I didn't see myself as an entrepreneur at all, but the environment saw me as an entrepreneur. Okay, when that was something very weird that happened to me. I got suggestions and opportunities to become an entrepreneur just from speaking with people. Yeah, so I'll give you an example. Tel Aviv University, very encouraging people, scientists to become entrepreneurs. So they have this program. Back then it was called Break Boost, that they give scholarships to PhD students, like a promising PhD students. And you need to apply and to have lots of tests and exams to get, because it's a big scholarship, and it's also they take you to a specific I would say conferences, and they give you a specific training. So it's a lot of investment, not just the scholarship. And they didn't apply to it; they just took me. Like they took me. I spoke randomly, okay, and I always say that entrepreneurship is not a profession. It's a character, okay. And I was so excited about my PhD project, even in the first year of the PhD, that I just spoke with anyone about it. Anyone wants to listen to me. So one day I was in the cafeteria or somewhere in the university, and I started randomly speaking with someone that just in the line of, I don't even remember how it started. I just, but that was very random. And she told me, I'm the right person to talk about with, because I'm the head of Brain Boost, and I didn't know anything about it. Okay, of a program that takes PhD students, and I will apply. Like, you got in, and that was like a part of the things that I said. That's interesting. And then I did this program, still thinking of myself as a scientist and not entrepreneur. But everything that I did in the lab ended up being a patent. And then Nurexon was established, and they called me to become the let's say CTO or to do the tech transfer from and I agreed. And exactly the same time, Shmulik just called me, said, I got your number and let's establish a company. So, and for everything I said yes, even though they were just, parallel. But I said yes, so I'm going to do this half a time, this half a time. I always say yes, so that's part of my,
I think your
that's
enthusiasm
part of
is
my character. First say yes, then think of what you are doing with this yes. But I always say yes, and that's how I end up just after the PhD. Like establishing Itay&Beyond and establishing Nurexon together at the same time. One is for spinal cord injuries with nanoparticles from the nose, and the other one is lab-grown brain tissues to find treatments for autism. And that's how it happened, because I just and it took me years to understand that I'm actually an entrepreneur. And what I like about it, being an entrepreneur and being a CEO, is not the sales or the money. It's the opportunity that I get to speak with people with passion of what I do, and everything else is a derivative of this. Like in Itay&Beyond, we have customers that are using our platform just not because I tried to sell them anything, because I spoke with them about the technology, and they just saw that it gives them a value. So my point is a different, is quite different. But I have to say that, and this is very important, as a CEO, you cannot afford to rely only on enthusiasm. You have to be also methodology, to work with methodology. To a business development, it's a profession. It's not, and it's not entrepreneurship. Entrepreneurship is what comes before. And I just found out that business development, it's a type of ability that you need to do it methodology. You not just by enthusiasm, but to speak with many people, to have follow up. And this is something I don't like to do at all. Okay, so for example, at Itay&Beyond, we had just recently a professional business development person that we work together, and he's doing the follow-ups. He's going to, to conferences and speak with people. This is his occupation. This is what he's doing professionally. And then when he have a lead or a very good conversation, I start to speak with them about as a CEO of the company. And I think this is basically, helping me to also understand about myself, what I am good at and what I'm not so good, or what is not my interest of doing in the everyday life.
Yeah, you need the passion and enthusiasm, but you also need the systems and the structure. There's no way around it if you want to build a successful business, I guess.
Yeah, I see myself as the person of vision. I can think of a technology, or I can think of a solution, and it may look at the beginning so as a fiction, or so far, and then I have this way to take it to the smallest parts, that if I do this and then it works, and because I know that this can be done, this can be done, this can be done, and then I think that the vision can work. I'm very good at this. But to start, speaking with people, I'm just losing patience at a certain time.
Totally. Yeah, it sounds like you're a procedural thinker, just like me. You like to think in processes. So yeah, same for me. And it sounds like you're super busy. So how do you currently protect your health and energy with all that you do? Like what habits make sure that you stay happy, healthy, and productive?
Habits. Okay, so first of all, I try to sleep. It's not. I will start from the beginning. I am busy, and not because I have few projects and I'm a CEO of the company, because I also have two small children. Okay, so that's a challenge, and to be involved father, like with children. And the CEO and the entrepreneur and everything requires lots of, I would say, concentration. And it's not exactly habit, but it's something that I became very good in, I think, is to be efficient, not to waste my time on things that I know that will just have too much energy of me. So to understand it, not I used to, at the beginning, I used to be very nice, and for example, answer any email that I get, even if it was like selling or so. I don't do it anymore. Not because I'm not nice, because it takes time that I don't want to spend. And I think that mental health is very important, and to separate. For example, I can tell you that I'm barely working from 4:30 to 8, around like this. Like, I don't reply email only if it's highly important. I don't take a Zoom call only if it's like something that is very, very urgent, because this is the time with the family, and it's very important for me. So I have my boundaries, and again, it's not like fully restricted, but it is. If I can take a call at 9 p.m. and not at 6 when I'm with my children, I will definitely do it. And it gives me boundaries. So I know that during the day I need to be very efficient, because my day ends at 4. It's not end at 8. Okay, so I need to be very efficient. And if I have something very urgent, I can do it at 9 after the children go to sleep. And try to keep the balance between family and work, because otherwise it's so easy to forget that you have other responsibilities. Very easy, and it's much more fun. At the end of the day, we need to be honest. It's much more fun to play with small brains in the lab, rather than to play with children in the garden. Like seriously, I love them, and like they're my life. Okay, but and I can be here for hours, but it's not the purpose. The purpose is to have a work-life balance and to really spend time with your kids and with your wife and with the family. It keeps your mental health very, very strong. And the physical health, you need to give up on something in life.
Oh, you can't give up on that.
I'm doing sports. I'm quite in shape, but I'm not as I used to. Not that I used to, and not as I want to be. Yeah.
You need to work on that. Well.
Yeah, definitely.
Last but not least, where can our listeners find you online if they would like to learn more about you or connect with you?
So first of all, I'm quite responsive in LinkedIn. We have it's the same name that we have here, Dr. Nisim Perets. In LinkedIn, we have the web and also Facebook if someone wants to take it more personal, I guess. But LinkedIn it's the way that I'm usually communicating. I also very active there. I share posts and updates on not only on Itay&Beyond, but also on things that I do in other projects as well. On also I think insights of entrepreneurship and other as a scientist also as well. So it's very interesting. Though not every day I post, obviously. Once in a few days. So LinkedIn, and we also have the website of Itay&Beyond, which is itay&beyond.com, or you can ask the ChatGPT. Also, Neuroscience is you can read about me in Neuroscience, and this kind of platform.
Wonderful. Nisim, thank you so much for sharing your fascinating work and insights with us today. It's truly remarkable work. And as I said in the beginning, it still feels straight out of a sci-fi movie. So keep up the great work. Take care and talk soon.
Thank you. Thank you very much. Bye-bye.
Thank you so much for listening to the Wired for Success podcast. I hope you've enjoyed this episode. If you did, please take a sec to rate and review my show or share it with a friend. That way you can support the show and help me reach and support other entrepreneurs like yourself as well. If you would like my help to figure out how you can go from overwhelmed and overworked hustler to empowered and confident leader, check out my website at www.wiredforsuccess.solutions and book a free 20-minute strategy session with me. Can't wait to help you get wired for success. Until next time, take care. Bye.